So it's a real pleasure to introduce our next speaker, of course, to introduce all of them, but particularly Makkuni Jayaram, who, as you can tell by his now gray hair, was my PhD supervisor a while ago. So he's going to speak to us about chromosome centromere. Thanks very much, Jay.
Thank you. Can you hear me at the back? Good. Yep. I'm going to discuss two DNA elements today, and I'll call one CEN, which stands for chromosome centromere, and the other is called STB, which stands for a stability locus element in a plasmid in yeast. So everything that I'm going to talk about this morning will be concerned with budding yeast, Saccharomyces cerevisiae. CEN is a critical DNA element on the chromosome responsible for equal segregation of sister chromosomes after DNA replication is completed. And STB is the partitioning locus of the yeast plasmid, which is responsible for the stable segregation, equal segregation of the plasmid after plasmids have been replicated. Turns out that this plasmid, which we will simply call the yeast plasmid, is a tiny circular molecule. It's only 6,000 base pairs long. It has about 60 copies in the nucleus, and you could think of it as a sort of selfish DNA element. It doesn't seem to do any good to yeast, and it doesn't do any harm at its normal copy number. And so it has evolved these mechanisms for extremely efficient partitioning and also stringent control of its copy number, such that at its normal copy number, it's an innocuous element, selectively neutral for its host. So here is the yeast centromere on the chromosome, and after replication you have two such centromere elements, and these CENs assemble protein complexes that then attach to spindles coming from opposite poles, and that's responsible for pulling the two chromosomes apart one-to-one fashion during cell division, so that every daughter cell gets one copy of the genome equivalent. Now, you can actually see this in yeast. So here is a yeast cell, and the chromosome, a single chromosome out of the 16 chromosomes, is tagged by fluorescence. And if you look, blank. That's interesting. Let's see. Yeah, there. If you saw that, the chromosome duplicates and is pulled apart by the spindle attached to the opposite spindle poles, so you get one chromosome in the mother compartment, one in the daughter. You can do the same thing with two chromosomes now, and you can tag one by red fluorescence and one by green fluorescence. And if you again do this experiment, time lapse, then you see one red separates from the other, one green separates from the other. So chromosome segregates sister to sister, one to one. Now if you look at the plasmid I told you also has a partitioning locus. And this we call STB. And normally there are multiple copies of the plasmid. Each copy replicates once per cell cycle, just like chromosomes. Just like each of the 16 chromosomes becomes 2 after replication, the 60 copies become 120, and then they are segregated to daughter cells. So if you look at plasmids, just the way we did with chromosome, again tag the plasmid with green fluorescence, you see multiple copies. These sort of form clusters. And these foci, again during cell division, you see they also split apart into the daughter cells, roughly equal segregation. Now that's 60 copies of the plasmid or thereabouts. So you can do the same trick as with chromosome. There are genetic tricks by which you can bring down the copy number to essentially 1 per cell. And so you can have one single copy plasmid labeled green and the other labeled red. And then if you do the segregation assay, you saw one green separate from the other, red separate from the red. So the STB locus-based partitioning functionally is almost identical to sen-based partitioning. Things segregate sister to sister. And so we'll talk now about the topology of STB and the topology of sen, because that's quite interesting. So eukaryotic centromeres are rather unusual DNA elements. So normally these centromeres are best described as long DNA elements, or very long or enormously long elements. And these are called regional centromeres because they occupy a significant region of the chromosome in size. And these usually are made up of repeated DNA elements. However, there is no consensus DNA sequence that's representative of regional centromeres. So the centromeres of the worm or the fly is quite different from the centromere of man, but they all are made up of repeated DNA elements. So these are not genetically defined in terms of DNA sequence. And these elements are epigenetically specified, that is, any DNA sequence can potentially become a centromere. And there are certain machineries that silence chromatin that are required for the epigenetic specification of these centromeres. So here is a typical example of a regional centromere at the top. So this is in the, So that's in the fission yeast, and the size of that centromere is about 75,000 base pairs long. And if it is a human centromere, the size is about a megabase. So that gives you the difference in the range of sizes of regional centromeres. And by sharp contrast, if you look at the centromere of the budding yeast, it's a genetically defined element. It's about 125 to 150 base pair. And there are 3 DNA elements that comprise the budding yeast centromere. Since it's such a short centromere, this is called the point centromere. So this is the regional centromere, a rather small one, and this is the budding yeast point centromere. So on each of the 16 chromosomes of yeast, the centromere is defined by this organization. The element CDE1 is short, the middle element 2 is about 100 base pairs, and the element 3 is about 2 turns of DNA, 20 base pairs. Okay, that makes up about 125 to 150 base pairs. So the point centromere has been a curiosity in centromere evolution. Where did it come from? And an evolutionary argument can be made, and we'll see if that holds up, that perhaps the point centromere of budding yeast came from a partitioning locus of the plasmid that it contained. So the functional part of that STB element is about 300 base pairs, and these 300 base pairs are made up of 5 repeats of a roughly conserved 60 base pair element. And if you look at this 300 base pair sequence, you can see 2 direct repeats of 150 base pair element, and so that matches the size of a single centromere, point centromere of budding yeast. Okay? So if you look at the sequence, both are rich in AT, but otherwise you cannot see this CD123 organization, at least in the STB locus of present-day yeast plasmids. It also turns out that this short centromere is present only in a dozen or 15 species of yeast, which all belong to a single lineage, a branch of evolution. And this branch point seems to be an ancestor species that lost somehow the machinery for making heterochromatin, either partially or completely. And I told you, for specifying these regional centromere, you absolutely need the machinery for laying down heterochromatin. Furthermore, this happens to be the only lineage where you see plasmid similar to the yeast plasmid that I described to you. Okay. So the point centromere emerged at the time that heterochromatin and silencing machinery became lost partially or completely in that lineage, and this is the only lineage that contains plasmids like the yeast plasmid. So the evolutionary argument has been that when the normal regional centromere function in this lineage was incapacitated, the plasmid STB was domesticated as a chromosome centromere and evolved the chromosome segregation pathway. Now, why topology? So we're trying to test this evolutionary argument by a topological test. Why topology? Because it turns out that the budding yeast centromere, the point centromere, has an unusual DNA topology compared to the topology of the rest of the chromosomes. So normally nucleosomes stabilize negative supercoil, so the genome is negatively supercoiled. But if you transfer the sen to a plasmid and maintain the sen in its active state or in its inactive state, and you can do that by genetic tricks, the active sen is more positive than the inactivated sen. So the explanation for that result, we'll come to that in a minute, is that the sen sequence, the 150 base pair sequence, which can arrange a single nucleosome, and nucleosomes at centromeres are peculiar because in those nucleosomes the histone H3 is replaced by an H3 variant generally called sen-PA. So normal histone contains H3, but the centromeric histone contains no H3. In its place, it contains a variant of H3. And this set of experiments by Steve Henikoff and Furuyama, the explanation was that the sen-H3, the sen-PA containing nucleosome, has a contrary right, that is right-handed rather than left-handed, or the other way around. Yes, it's right-handed DNA rather than left-handed DNA. Solenoidal right. I don't know whether that explanation is correct, but if the active form of SEN engenders positive supercoiling, and if STB and SEN are evolutionarily related, we could test whether STB is also positively supercoiled in its functional state and not so in its non-functional state. So that's essentially the test. And so here is how we do this experiment. So we construct a plasmid in which we have a copy of STB, and we also have a copy of a yeast centromere from chromosome 4. The idea is that you have an internal control here for the topology of SEN for reference. And then we flank the SEN sequence by 2 direct copies of a recombination site-specific recombinase. And in yeast, the nice thing is that you can arrest cells in G1 phase of the cell cycle, that is before DNA has been replicated, and induce the recombinase at this point so that you get the SEN circle excised from the parent plasmid. So you have a circle that contains STB and a smaller circle that contains just the SEN, right? And then you can look at the topologies of these individual circles. And in this basic experimental setup, you could inactivate SEN or inactivate STB or inactivate both and ask how the topology changes. So this is essentially the experiment. So here is the parent plasmid, this one, and after recombination, this is that plasmid, and this tiny circle is someplace here below. Okay, so quantitatively, you excise these 2 circles, then you release the cells from G1 arrest, so DNA replicates, and then you can arrest the cells in metaphase after replication is completed, and you can do the topological analysis. So this is the assay. So here is the STB circle, and by genetic tricks we keep the functional normal STB, and here is when this STB is inactive. And then you display the individual topoisomers by standard chloroquine electrophoresis, and you can see that the centromere, the STB topology distribution is centered some point here in the functional state, some point here in the non-functional states, and the linkage difference is between 1 and 2 units, closer to 2. And it turns out that 2 is the linking difference that was reported for active SEN versus inactive SEN. So qualitatively and quantitatively, the topology of a functional SEN and STB versus their non-functional equivalence is the same. Yeah? And here is the topology of the SEN circle that came out of here, and you don't see any difference whether or not STB is active or inactive in the plasmid. That's what you expect in this case. Both SENs are functional. Okay? So you could inactivate the STB locus in a number of ways. So in the set of experiments that I showed you, we inactivated it by depriving it of the partitioning proteins normally that the plasmid makes, which is required to keep it active. We also know that in G1, when the cell is not gone through replication, prior to replication, the topology of STB is different, and the STB is in a non-functional state. So the partitioning clock is reset at the G1 to S transition state of each cell cycle. We also know that if you depolymerize the spindle, the thing that pulls chromosomes apart, then the partitioning system or the plasmid is also non-functional. And you can depolymerize the spindle by using a drug called no-codesolvent yeast. So here is functional STB again, and here is two non-functional states of STB made by completely different methods, one before replication, one by spindle depolymerization. And you can clearly see that the center of distribution, the functional STB versus the non-functional STB, is again shifted towards negative. So the functional STB is more positively supercoiled than the non-functional STB. And again, in the Sen circle here, there is no difference in topology, right? Because the Sen is active in all three cases. Now here is sort of the penultimate experiment. So here we have the same plasmid, which has STB, 1 copy, and Sen 4. And the experiment is again the same. Arrest the cells in G1, and then do what you want to do, and then release the cells. So here what we are doing is either inactivating Sen by a specific mutation, or inactivating STB by depriving the partitioning proteins, or doing both in G1, and then releasing the cells, and then assaying what the topology is. The difference here is that we don't resolve the Sen and STB into two circles, right? So if quantitatively the topologies of the two loci are the same, you should see the additive effect. So that's the experiment here. So when you have both the loci active, you have the distribution centered around here. You have Sen inactivated, it shifts to negative. When you have STB inactivated, it shifts to negative. When you have both inactivated, you get double the shift, or nearly double the shift. Okay, so not only qualitatively, quantitatively, the results match. In fact, the Sen STB topologies are additive when they are on the same plasmid. Okay, so now to make sure that's true, we did exactly the same experiment. This identical experiment split into two parts, and in one case we resolved the two circles. Right? So in all the experiments so far with the two resolved circles, we had kept Sen active, so we did not see any shift in the topology of the small circle. Now here is STB is plus and plus, and when it's minus and minus, you see the topology shift in the two lanes to the right towards more negative. And now here is the small Sen circle, plus, minus, shifted more negative, plus, minus, shifted more negative. So the composite topology that we measured in the last experiment with the big plasmid is equivalent to the sum of the topologies that we now measure in the resolved two circles. Okay, so I'll give you one final experiment, and this is the same STB plasmid with no Sen, but we integrated that sequence into a yeast chromosome, and then flanked that integrated sequence by two recombination sites. So to start with, you have a single chromosomal copy. So the question is, if you move the STB from plasmid to chromosome, if you transplant it to the chromosomal context, will it still show the topology? So in G1, you arrest the cells before replication, induce the recombinase. So you get this circular piece of DNA out, which is the same size as the previous circle. You have a copy of STB. So this is the topology in the integrated state, and this is the topology of the Xi circle. Again, the shift is more negative. This is without the partitioning proteins. So when you have a functional STB on the chromosome, you see the same difference in topology linking number, which is shifted towards the positive side when the STB is functional and less positive when STB is non-functional. So the sum of the experiments I told you, described to you, is that SEN and STB have a net positive supercoiling, and one might argue whether this is because of the wrong-handedness of the nucleosome. That's still debatable. However, clearly there is a topological signature that identifies the chromosome partitioning locus and a selfish DNA that's dependent on the host genome partitioning locus by positive topology. So this adds another topological, tantalizing piece of circumstantial evidence to a long list that seems to suggest that the short centromere, unusual short centromere, the partitioning locus of the chromosome and the partitioning locus of a parasite plasmid must have had a common evolutionary origin. I'll stop there. Andre.
What about the number of nucleosomes? Have you analyzed that there is the same number of nucleosomes, or
in the two plasmids?
Yes.
Yeah, I mean, so this was the argument made for the SEN case, that the idea was that if you have a normal nucleosome, right, and you have the unusual, the histone variant containing nucleosome, you have a topological change of roughly 2. So by that logic, if you inactivate, you lose this, and that comes off, and this goes on, and that's how you get the linkage. And so, and not because, you know, this is not occupied, the sort of argument, number of nucleosome argument, not because this was initially 0, and when you have the inactivated state, 2 nucleosomes, normal nucleosomes, get on. So very cleverly, I mean, this I'm just describing to you the Henikoff experiment. They shortened the plasmid such that there was no room to load the 2 nucleosomes, and yet they got the topology shift. So their simplest argument was that here is a shift in the sign of the DNA handedness, the right to the DNA around the nucleus. But the trouble is that, you know, almost every experiment that has been done since, both in vitro, crystal structure, it seems that the SEN-PA nucleosome organizes a normal octamer with the usual left-handed wrap. So the question is, should we explain the in vivo topology in terms of a wrong-handedness of DNA, or is it something else that permanently fixes the positive topology to SEN? I'm tending towards the latter explanation. However, what we are trying to do now is to excise the minimal STB as a small circle so that there is no differential topology involved here. So whatever nucleosomes are assembled will be on this small circle. You do not get any of the normal left-handed nucleosomes from the rest of the plasmid, and see if we can get the absolute topologies of these circles and measure the nucleosomes present on those. If we can do that, we are trying to do that, then this debate about whether there is wrong-handedness of DNA or whether this topological feature is frozen at the centromere as a topological message that the partitioning complexes, should it be assembled there, can be solved. I don't know the answer. Yeah, but it was the length argument that argued for the handedness. Yeah.
Because still, sorry, there are also other proteins known to bind to DNA, which are kind of par proteins, which and some of them change the DNA helicity.
Sure, it's entirely possible. Again, you know, I have to, since we haven't done the single STB topology, I have to rely on the Henikoff argument. that they made another mutation in the centromere proteins, one that binds to the spindle so that the stretching force of the spindle is not an effect. And they still got this topological. So they were trying to put forward the simplest possible explanation. Yep.
So I guess many groups believe that there's nucleosome positioning sequences in DNA. So with regular nucleosomes, it's something about the DNA which predicts where the nucleosomes sit. So now you have a variant.
Yes.
And there's a variant in the nucleosome in one of the histones. But 150 base pairs is sort of, it's I suppose it's the length on the nucleosome, but it's also a persistence length. Do you think there's something special about the DNA sequence which contributes to the left and right-handed?
Yeah, so the point centromere, which I told you is present only in budding yeast and a few members of that particular lineage, they all have this 1, 2, 3 DNA elements with some slight variations. So you need that first element, which is about 10 base pair, the second, which is an AT-rich sequence, which is about 88 to 100 base pairs, and the third element. And this is genetically defined. So unlike the other centromeres, when we talk of centromeres, you're essentially thinking of these long regional centromeres with repeat sequences where silencing machineries assemble and so forth. So the general thought about regular centromeres, I think, will not apply to point centromeres. So in yeast, you've mapped centromeres, the nucleosome positioning with respect to centromeres. The CenPA, the specialized nucleosome, is centered about the middle of this 1, 2, 3 consensus element. And that is specified by proteins that first bind sequence specifically to those elements.
So the question is, do you believe there's something mechanically special about the DNA sequence that contributes to this?
I mean, the mechanical specialty that you're thinking of might come from the initial binding of the scaffold protein in a sequence-specific fashion. And that might induce the mechanical flexibility or specialty that you need to recruit the other proteins to assemble the, yeah.
If there is an evolutionary relationship between these two systems, wouldn't you argue that some of the proteins must be conserved?
Can you repeat the question, please?
The question is, if the plasmid, I'm trying to paraphrase the question. If I'm wrong, please correct me. If the partitioning locus and CEN are evolutionarily related, won't you see functional similarity in terms of the proteins that each of these locus assembles? So in other words, is the chromosome partitioning complex the same as the partitioning complex of the plasmid? The answer is no, clearly no. But strikingly, for example, the plasmid doesn't assemble the kinetochore complex or attach to spindle and segregate by the spindle mechanism. But you do see the binding of several chromosome segregation factors, proteins that bind to centromere. For example, chromatin remodeling complexes that bind to centromere bind to the STB locus. The cohesin complex, which is required for segregation of sister chromatids, also bind here. And the CenPA protein, the nucleosome, the H3 variant, also binds to STB. Whether it assembles a CenPA nucleosome is questionable because the stoichiometry of binding at centromere and binding at STB is very different. So one might make the argument, let's say that these are in fact evolutionarily related partitioning loci. Even though the plasmid, as I said, is selectively essentially neutral, having 60 copies of it still has a certain penalty, fitness penalty. So if you do a long-term competition experiment between a strain that contains the plasmid and one that does not have the plasmid, the one without the plasmid wins eventually. Okay. So if both chromosome partitioning and plasmid partitioning started off as the same, there would be some pressure for the chromosomes to evolve a different pathway to get rid of the evolutionary burden posed by the plasmid. The counter-evolution of the plasmid was to evolve some mechanism by which it can still segregate like the chromosome. So the reason why some of these chromosome factors still bind to STB locus might still be, looked upon as evolutionary vestiges of their common kinship. Functionally now, the plasmid partitioning pathway and the present chromosome partitioning pathway have diverged sufficiently. Chromosome trying to get away from the plasmid as a parasite element trying to maximize its partitioning efficiency.
And if you look at the most recent experiments which suggest that the nucleosome structure of CEMP changes during the cell cycle, how does your model fit with that?
Yeah, I mean, it's consistent. So, for example, when you do not have CEMP, it turns out on the yeast chromosome, CEMP is loaded at the G1 to S transition phase of the cell cycle. That's before replication or at the time of replication, and then stays on through the cell cycle till the next replication event happens on the chromosome. In the plasmid, the CEMP associates at the same time, G1 to S window, then stays on through replication, and after the segregation is completed, it comes off the plasmid locus before it comes off the chromosome locus. So there is a timing difference in when the dissociation occurs, and that's why when you look at G1 stage, I told you the plasmid STB locus is more negative in supercoiling. And once you go through replication, CSC4, the CEMP associates. So we think that CEMP association is critical for this positive shift in supercoiling, but whether that's because a nucleosome with a right-handed wrap is formed is questionable.
I think we have
Did I answer it? Yeah.
I think we have time for one last question. Maybe Ian?
You said that the STB locus is 2 direct repeats of these 150 base pair elements.
Roughly, yes.
Is a single element functional, and does it have the same change in linking number as the 2 elements?
Yeah, so the single element linking number shift we are looking at, I haven't done that yet. However, if you make deletions of those repeated elements, you can, so there are roughly 560 copies of the consensus element. If you remove 2 to 3 of them, you still get partitioning efficiency, which is roughly the same as normal. But if you go to 4, you lose partitioning efficiency. So 150 is about the limit of the minimal STB. That would fit in with the argument. Thank you.
Let's thank Jay for a great talk.